Okay, go right ahead, Ambika.
Great. Hello, my name's Ambika Grover. I'm a senior from Greenwich High School, and I'm very excited to be sharing with you my research entitled Design of a Novel, Dual-Functioning Tissue Plasminogen Activator and Factor XI Inhibiting Anticoagulant Therapeutic for Rapid Ischemic Stroke Treatment. So strokes are the second leading cause of death worldwide, affecting 15 million people each year. Of this implicated group, there was unfortunately a 5.5 million person mortality rate, and an additional 50% of survivors are left with chronic disabilities. Now, there are two types of strokes quite largely. The first is an ischemic stroke, and the second is a hemorrhagic stroke. A vast majority of strokes, or 87%, are classified as ischemic, and in this situation, an artery narrows or becomes wholly blocked, which restricts the movement of normal blood flow and deprives the brain of oxygen, glucose, and other valuable nutrients that are essential for homeostasis. Now, there are also two defining important features of clots to note, which are platelets and fibrin, and this is what is going to accumulate in the artery and cause a blood clot to occur. So tissue plasminogen activator, or TPA, is currently the leading treatment for ischemic stroke. It was approved by the FDA in 1996, and since then we haven't had a very substantial or new kind of drug that has been approved. And the way it operates is that it catalyzes the conversion of plasminogen to plasmin. And plasmin is basically responsible for breaking the cross-links between fibrin molecules, and keeping in mind that fibrin is one of those defining aspects of a clot to begin with. So while TPA is this gold standard sort of treatment, there are several associated risks and shortcomings with TPA. The first is that there is free circulation throughout the bloodstream. It also poses the risk of something called reperfusion injury, which can cause further bleeding in the brain, and it also lacks thrombus specificity, and a thrombus is basically just another word for a blood clot. So as a result, TPA is used less than 10% of the time, even in emergency situations. But there's also another challenge that existing research has not combated, which is something known as the coagulation cascade, and that is the mechanism by which a clot is actively rebuilt. And current research hasn't really found a way to deter this as well. And so these sort of challenges in not only TPA, but also in its limitations into actually realizing the full extent of ischemic stroke treatment set the framework for what I wanted to engineer and resolve through the work in this sort of research I'm going to share with you. So the first place to start is with the coagulation cascade. And as I mentioned, this is the mechanism by which a clot is actively rebuilt, and it involves a series of factors that basically accumulate to create fibrin and generate that very secure blood clot that can be quite harmful. Notably, it consists of the extrinsic pathway and the intrinsic pathway. So when you get a cut, for example, the intrinsic pathway is your platelets activating to close it up, but the extrinsic pathway is this really harmful pathway that results in unnecessary accumulation. So after realizing that this was a huge challenge that really no treatment was aiming to simultaneously stop and resolve, I researched a new sort of way to sort of resolve this in my therapeutic. So I found that a vitamin K antagonist known as dicumarol is a naturally derived compound found in sweet clover, and this targets several of the clotting factors as indicated in this diagram. Notably, I actually ended up changing the anticoagulant I used in my research a little bit later on, but this set the initial framework for how I wanted to stop this cascade as a part of my therapeutic. So this first part of the methods and beginning this process of synthesizing this nanoparticle treatment to be administered intravenously was the synthesis of interior dicumarol Fe3O4 nanoparticles. So Fe3O4 nanoparticles have several advantages for use. The first is their magnetic response, their potential for external control and guidance. So in case we need even further localization to the site of the clot, that is something we can do. And there's also the ability to add additional layers without losing any of these desired properties. So to conjugate these nanoparticles, I engaged in a multi-step process consisting of repeated sonication with different things. For example, I started with chitosan to build this initial layer, then I added dicumarol, and then sort of sealed that again with chitosan. And this sort of concisely illustrates that process. And as I was doing this, I was able to verify the shape and size of the nanoparticles through a few different measures, including a scanning electron microscope, dynamic light scattering, and confirm the structure was actually changing through ATR-FTR spectroscopy as seen on the right of your screen. The second part was the fabrication of the SiO2 TPA component. So the first part was, let's create this interior aspect that's going to deter the coagulation cascade. But the second was finding a biologically inert and settable substrate I can embed TPA into. And this was a secondary layer to encapsulate those nanoparticles. And so as you can see here, I was once again able to verify this conformational change was occurring through ATR-FTR spectroscopy. And then this is sort of a quick illustration what the second layer actually looks like. The third part was to use dynamic light scattering to verify that these were actually increasing over time. And so the way I did this was I basically measured the different nanoparticles, the first of the Fe3O4 nanoparticles, the second with the dicumarol-coated nanoparticles, and the third with the SiO2 TPA nanoparticles. And I saw that size change from about an average of 20 nanometers to 40 nanometers, which verified that I was in fact seeing a change and this cumulative layer building was in fact happening. From there, I decided I wanted to create a different kind of structure to ensure that this therapeutic could be administered in the most efficacious way possible. And for this, I chose a microbubble structure, which has been used in various applications for different kinds of therapeutic for different diseases. And the reason why this is so particularly effective is not only because it succinctly encapsulates different things, but also because as it's being administered, we can use something called ultrasonic vision to actually see where these are going in the body in real time because of their unique structure. And so to conjugate this particular structure, I added 75 microliters of SDS and then combined these nanoparticles I'd previously created to create these porous like microbubble structures, and then used a scanning electron microscope as seen on the right of your screen to verify that I had indeed created these structures. And each microbubble consists of a spherical shape with an average diameter of about 200 micrometers. So furthermore, the last kind of component and the thing I wanted to resolve in my research was figuring out how do I make this therapeutic specific to the site of the clot. And so keeping in mind, there are two key parts of clots that I mentioned, platelets and fibrin. And so what I did after researching and looking up at different signatures to these both platelets and fibrin, I found that integrin alpha 2b beta 3 is expressed in very high levels in platelets and their progenitor cells. And I found specific peptides that would both be selectively binding to platelets and fibrin specifically, and conjugated them with corresponding malamides, which basically completes the synthesis of peptides so they can now be attached to the external of the microbubble. And I used ATR-FTR spectroscopy to verify that change over time. And on the top of your screen, you'll also see what those nanoparticles were looking like as they were being sonicated into the microbubble structure. So to give you a quick overview, as there are a lot of moving parts in this therapeutic, there's first this interiormost layer, which consists of these iron oxide nanoparticles coated in the anticoagulant. We then have a secondary level of SiO2 TPA. And then finally, we have this microbubble structure finished off with peptides that are going to be very specific to the site of the clot. And so how this might function in real time is that platelets are activated by endothelial cell dysfunction and the coagulation cascade would begin. And these designed microbubble structures can bind to the thrombus and release TPA followed by an anticoagulant. So I used two verification methods to ensure that this therapeutic I built was in fact going to be effective. The first was by creating in vitro clots in the lab using a vertical gel system. And so these vertical channel gel systems are composed of fibrinogen, thrombin, and agarose. So agarose is this gel-based medium we're able to create, and fibrinogen and thrombin create this white thing you're seeing on your screen, which is fibrin. And the significance of this is that the white area you're seeing is fibrin, and over time, the increasing amount of clear area, or that white area that's becoming more and more muddled, is indicative of a clot area that's being lysed in real time. And so what I did is I took 15 microliters of each treatment, which were saline-free TPA, SiO2 PA, the microbubbles with and without the peptides, and added them to the top of each vertical channel gel system. And these gels were incubated at 37 degrees Celsius and observed over 12 hours. And the results were quite promising because the microbubble containing the peptide resulted in a 20% decrease in the fibrin dissolved, which is shown in blue, which was twice as high of that of TPA alone. So this initial verification component was quite promising. But from there, I actually wanted to create real blood clots and examine how this might work. So I got my own blood drawn and I used something called bleed stop, which is a coagulant used for wounds, and emulated these in the lab. So the way I did this is I created these little bleed stop molecules here, put my own blood in it, measured the absorbance, and created these halo blood clots added the therapeutic, and then observed the post-absorbance. And what I found is that the treated blood had the most significant absorbance, which was indicative of a liquefied clot, as you can see here. So that halo blood clot essentially became liquefied once more, which represents the idea that an artery is now having further blood flow. So to also verify that this would be effective in blood flow conditions, I imitated the circulation of blood real time in the body, which is at about 10 centimeters per second. So that's how the rate of arterial blood flow. And I basically circulated these microbubbles through a pump at this period and measured and basically wanted to ensure that the structure was being maintained at that point. So this you'll see on the right of your screen, and on the left of the screen is a setup of how I actually mimicked this. So this is where I also realized after conducting this initial portion of my research, I talked to a lot of leading ischemic stroke doctors in the field. And one of the parts of our conversation I had was that dicumarol is not going to be effective in some instances. So this initial anticoagulant I'd picked was not necessarily the most optimal in some instances. So after doing some further research and looking into what people were doing in terms of anticoagulant treatment right now, I found that factor 11 was really critical in this new research that would allow you to stop a clot from building without any of the associated bleeding side effects. And the reason why this is the case is because factor 11 is at the intersection of the intrinsic and extrinsic pathways, which means it's not associated with something that is essential to stop bleeding in the first place. So for this, I selected a factor 11 polyclonal antibody as a replacement for dicumarol. And what I did is I took a factor 11, the factor 11 antibody as you're seeing here, and verified that after exposure to this polyclonal antibody, factor 11 structure is hindered and thus stopped in that regard. And that can help us in the sense that it verified I can use this as an anticoagulant that will be not only more safe, but also equally as preventative in its measures. So just to wrap up really quickly, my research was able to design a new targeted and dual functioning therapeutic as a new method to treat strokes. And through its dual application methods, we're able to resolve two of these challenges. But the potential applications extend to other thrombus diseases, including deep venous thrombosis, arterial thrombosis, and heart attacks, which means that this can extend to a lot of diseases associated with blood clots. So I want to just first quickly thank everyone for this lovely opportunity. I'm incredibly grateful. And these are some of the people who I had wonderful conversations along with the way: Dr. Zecchi, Dr. Massey, Ms. Muller, and Mr. Bramonti, my mentor. Thank you very much.
Thanks, Ambika.